Next, I'm happy to welcome Erik Manting, CEO at Immunicum. Welcome, Erik.
Thank you, Michael. So in the next 15 minutes or so, I will guide you through our corporate presentation. It's on our website. If you'd like to look into more detail, I will just give you the highlights today. Let's start with the bigger picture in immuno-oncology. For a long time, it was believed that the immune system had no role in controlling cancer, or at least not in the therapeutic setting of controlling cancer. And the reason is that, first of all, of course, there was already a tumor established, meaning that there was no longer immune control. But also, very importantly, chemotherapy, of course, does affect the immune system. And chemotherapy is applied a lot in cancer, so the general thinking was the immune system is so disrupted and is so brought down by the chemotherapy, it will no longer have an effect on the tumor. And then, roughly 10 years ago, there was a breakthrough in the sense that immune checkpoint inhibitors, a specific class of antibodies, showed to actually take the brake of the immune system, and in a number of tumors that resulted in the immune system basically providing control over the tumor. So that was a big breakthrough. After that, thousands of trials were launched with a lot of different immuno-oncological approaches, but unfortunately not with a real significant shift in terms of the benefit of the checkpoint inhibitors. So a minority of patients is responding to checkpoint inhibitors. Some tumors do not respond at all. But also there has not been really big steps forward in combination with these checkpoint inhibitors. So something needs to be rethought in a fundamental way. And that's one of the things we are currently doing. So next to our technology basis, we have also thought about how do we position ourselves in the very competitive immuno-oncology landscape, given the historical basis of where the field has moved. We believe that there are two elements in today's immuno-oncology that we can use to position ourselves most competitively. One is tumor recurrence. Today, 90% of patients that die worldwide of cancer die from tumor recurrence and not of the primary tumor. So it's a big unmet medical need. And what we are trying to do actually is to develop therapies that prevent tumor recurrence, which is of course very different from waiting for a tumor to recur and then try to treat the tumor, which is then very often much more difficult to treat. So the prevention of tumor recurrence, we believe, holds a big therapeutic benefit, and also we believe we have products that can address that medical need. And secondly, we focus on those tumors that are classically poorly responding to existing therapies, including immuno-oncological approaches. That's what we call hard-to-treat tumors, and what we try to do there is that we plant dendritic cells, which have as a function to basically activate the immune system, and we plant them as a red flag into the tumor, hoping that it will break the immune tolerance of that tumor and hopefully to better outcome of the treatment. We have a lead program in AML, which is in phase 2, which is reading out throughout the course of this year. That product is an off-the-shelf product, so it's made from a cell line, and that allows for centralized manufacturing, which is also an important lesson learned in the industry, that cell-based products are difficult to manufacture. But if you start with a cell line, it's much more predictable and also much more scalable. And off-the-shelf means that we freeze the product so that it is available on demand for the treatment of the patient. And then finally, we have a number of additional clinical trials. We are developing the intratumoral primer in gastrointestinal stromal tumors, and we are also running a trial aimed at the prevention of tumor recurrence in ovarian cancer. We have a strong R&D organization. It is based in the Netherlands, in Leiden, and that research team not only supports our clinical pipeline, but also helps to develop and further optimize our manufacturing processes, and of course will fuel the longer-term pipeline expansion. And as you know, we are listed on the Nasdaq Stockholm. So as a summary, the AML study is called the ADVANCE2 study, is now in phase 2. It's fully recruited. We just provided the data update that I will tell more about, but also we expect further data updates this year. The TROY study is the study in GIST that we are preparing for. As a next step, we did do a smaller trial in GIST, but we are looking for a signal confirmatory trial, and that will be a next clinical study. And then finally, the Allison study is the ovarian cancer trial that I mentioned. The biology of our products relies on a specific insight in dendritic cell biology. Dendritic cells are talking to each other, which you call crosstalk. And also they exchange antigenic material, which is basically triggering the immune system towards the tumor. And we have done a lot of work with our own products on that, but also more recently it has been surfacing that for the triggering of immune responses against tumors, it's actually a very critical pathway. So we are on the one hand in the middle of that field, but also because of these indirect priming mechanisms, it allows us to develop allogeneic products, again, products that have a much better manufacturing than products that are derived from patient material. So on the one hand, these pathways are very important in the effective triggering of immune responses against cancer cells, but also a very big advantage is that we can use this to design allogeneic products. DCP-001 is a vaccine that we apply intradermally. It's derived from a cell line, the DC1 cell line, which belongs to us. It's a proprietary cell line. We've also developed our own manufacturing process. And Ilixadencel is derived from donor material, meaning that we start with leukocytes from healthy donors, and then we isolate the cells that we need to make in the end the product, which is something we administer in the tumor itself, so intratumorally. And then we have a broad pipeline that we are not only building up ourselves, but also in collaboration with a broad academic network and also with selected other companies. Now let me start with DCP-001. Why does this have a competitive product profile? On the one hand, of course, we are addressing a high unmet medical need in what we believe in a very unique way, the prevention of tumor recurrence. But for that you need not only a product that is very immunogenic, and that makes it potentially very effective, but also it needs to be very safe. Because if you try to prevent something and patients have already undergone initial treatment, those treatments should not harm the patient's health or the quality of life. So that it is a safe product is also a very important part of our approach. It's administered as a simple administration via intradermal administration. And because we trigger an immune response, including immunological memory, that leads to a longer control over the disease. And then finally, of course, off-the-shelf convenience, meaning that the product is available on demand for treatment. What we saw in the phase 1 is that in AML, because it's such aggressive disease, patients immediately become ill, and these were patients that actually were already no longer in a remission, so no longer clinically healthy. When we started the vaccination, that's the yellow line on the left, and you cannot stop that disease with an immunological approach. But what we did see, and that was very interesting, is that the patients that were in a complete remission, that had a low level of cancer cells remaining, showed a very good response to the therapy. Within the end, a relapse-free, so disease-free survival of 14 months and a median overall survival of 36 months. So this was the basis to pursue this program. And what we have now added, and this is an additional element to the AML field, is measurable residual disease, or MRD. So nowadays, with sensitive molecular techniques, you can measure if a patient still has residual cancer cells or if the disease is completely gone. And the patients that are MRD positive, that have residual disease, they have a very poor prognosis. So we focus on that patient group. We focus on patients that are in a complete remission so that we can do the vaccination. And then, of course, what we hope to do is to prevent or delay the relapse and basically provide for a prolonged remission, so a prolonged disease-free status of the patient. Why MRD is so important is also shown in this historical analysis. It's a historical analysis involving more than 11,000 patients. And what you see is that there's a dramatic impact of MRD on the survival curve of these patients. And also, as you can see in the quote of Dr. Uwe Platzbecker, with whom we collaborate, again, safety, not impacting the health and quality of life of patients is very important to develop maintenance therapies that are addressing this challenge, so keeping patients alive without disease longer. This is a product that has recently been developed, actually the trial, and this is a study from Bristol Myers Squibb, a big pharmaceutical company. It was run in parallel to our trial. This drug is now on the market. It's an oral chemotherapy called oral azacitidine. And what you see is that in patients with residual disease, if you don't treat them, and that's the placebo group, which is represented by the gray line, there is a very steep drop-off of patients that are still disease-free. And actually more than half of the patients is ill again within the first 3 months after initial treatment. With azacitidine, you shift that curve to 7 months. So it got the drug approved, but nevertheless, it also shows that there is still a lot of room for improvement. What we are doing is we are applying a vaccination scheme of 4 biweekly vaccinations, and then after 2 months, another 2 booster vaccinations. And then we look at what it does, first of all, on the level of MRD. And this is data we reported recently. This is the complete 32-week MRD data of all the 20 patients that we included in our trial. And what you see is that 14 out of 20 patients responded. So either with a complete conversion of their MRD status, and those patients that converted remained relapse-free and alive during the follow-up. But then there were also patients that showed a strong reduction of MRD or that were relatively stable with MRD. And unfortunately, 6 patients were already, again, getting sick and immediately got the disease back. But that's a relatively good start. And what we could also show is that actually the relapse-free and overall survival estimates that we can do, so the good news is they have not yet been reached. We have followed up these patients now for a median of 14 months, and relapse-free survival median has still not been reached. So that's first of all a good and important fact. But we could do an estimate of the amount of patients still alive at 6 months. And for relapse-free survival, that's 83.7%, and for overall survival, 97%. So this bodes very well for the continuation of the trial and the readout we expect to report on in Q4 of this year, when we will have all the data in the 72-week study period for all patients. But this was the first data that we presented 2 weeks ago, showing that the effect on MRD also has an effect on and a relevant survival benefit for these patients. As a very short summary of the AML treatment landscape, because it's very broad, we focus on those patients that are undergoing high-dose chemo, which is roughly half of the patients that are diagnosed with AML. And then patients either get a transplant or they get nowadays oral azacitidine, which has now been approved. So very logically, we will focus on that patient group that is treated with oral azacitidine and then see if we can combine that with our vaccination approach. To support that, we've done a lot of preclinical work, including this mouse model, which is a humanized mouse model with a human tumor and a human immune system, where we could show that the vaccination actually can be combined very well with azacitidine and also with other drugs like venetoclax, and it has a synergistic effect. So we have done the preparatory work for a next trial to be combined with azacitidine or venetoclax or both as they are becoming a current standard of care. I will go a bit quicker over the next set of slides. So what we are now trying to do in ovarian cancer, where also initial treatment is very often successful, it's chemo combined with surgery, but the disease comes back in the majority of women very quickly. So also here what we try to do is do a vaccination to see if we can prolong the disease-free status of these patients. That's a phase 1 study, so we will mainly look at safety and feasibility and immune responses against the vaccine, but of course also looking for initial clinical data with respect to potential disease-free survival benefit. The setup is the same, so 4 vaccinations followed by 2 booster shots. We expect somewhere halfway this year, maybe end of the second quarter, beginning of the third quarter, that we can report initial data from this trial. And then finally, on the intratumoral primer Ilixadencel, it's healthy donor-derived dendritic cells that we manipulate in a certain way so that they become very immunogenic. We inject them into the tumor. And we have done a lot of clinical work with this in a lot of different indications. And the main thing we have to decide is what can we now take as the best indication moving forward. The reason we chose GIST is that after first-line treatment failure, there's very little success with everything which is currently available. So there's a big medical need to treat that disease after initial treatment fails. And what we have shown in a previous study is that in patients that had already been heavily pretreated, we could restore responses in 2 out of 6 patients. So what we need to do now, because this is relatively small patient numbers, is to do a so-called signal confirmatory study, which is what we are preparing for, to confirm that we can have a significant impact in GIST. Now that leaves me with the management team, to which we recently added Leopold Bertea as a Chief Technology Officer. He comes from Novartis, he has a lot of experience with cell-based therapy, so he will help us establish manufacturing infrastructure. And then also, of course, mentioning our shareholders. We currently are supported by the strongest shareholders we can hope for in both the Netherlands and in Sweden, so Van Herk Investments in the Netherlands and AP4 in Sweden, and also as management we have acquired a significant amount of shares. Finally, as an investment summary and outlook, we are addressing, we believe, key challenges in the cancer therapy landscape. We have now a focused clinical pipeline with multiple near-term milestones, a competitive product opportunity in AML maintenance, with more data on MRD, relapse-free survival, and overall survival shaping up in the course of this year. A first step into another indication, being ovarian cancer, and preparing for a signal confirmatory trial in GIST. With that, I would like to thank you for your attention, and back over to you, Michael.
Thank you so much for your presentation, Erik. Have you seen increased interest in your products lately?
Yes, I think what we initially saw, Michael, was a very positive response from the hospitals we work with, because these doctors deal with this disease on a daily basis and they want to treat MRD. And the fact that we did see initial responses on MRD and also in combination with a product with a very good safety profile, that led to initial optimism also at our end, of course. And what we now start to see is that for the first time we can start reporting on how the effects on MRD start translating into survival benefit. And that is something that for, let's say, people that are less specialized in MRD, for example, is a more easy to understand phenomenon. And of course, we're very happy with the data that we could report 2 weeks ago, and that are further shaping up in the rest of the year, because also we want to see, of course, that the effects on MRD translate into a relevant survival benefit. So now I think we have a good basis to have more interaction with external parties like investors and pharmaceutical companies, because they start to see now what the potential of the drug is.
That's great. Next, I wanted to ask you about, well, specifically the fact that your candidate is a cancer relapse vaccine. There's been a little bit of hype around cancer vaccines lately, and I'm wondering, how does your candidate differ compared to the competition, let's say?
I think it's an excellent question, and there's two sides to the answer. One is that the immune system is really suffering from cancer. So when there's established disease, tumors have the tendency to suppress the immune system. And we believe that's also one of the reasons why it has been so difficult to really have a big step beyond what we have seen with checkpoint inhibitors. It's very difficult once there is established disease to trigger relevant immune responses. So that's one of the reasons we focus on tumor recurrence. What you see after initial chemotherapy is that there is a suppression of the immune system, and then there is a sort of reset of the immune system. And we believe that that offers for a good window for vaccination, because you have basically gotten rid of all the cells that are suppressing the immune system, including the cancer itself. The other side of the answer is the design of the vaccine. As you may remember, we basically trick cancer cells into looking like dendritic cells. So we have a proprietary cell line, it's a leukemic cell line that we can grow in cell culture, but that is not very immunogenic. But by forcing them to look like a dendritic cell, they become very immunogenic. Right. But also they are still cancer cells, so they carry what you call endogenous levels of tumor-associated antigens. So because they are cancer cells, they are naturally expressing all kinds of antigenic material that the immune system recognizes as a tumor. And that makes it very different from vaccines that are only focusing on selected antigens, for example, and that are based on, for example, peptides or more specific compositions that will trigger immune responses. But we believe that there is a specific side to working with a whole cell-based vaccine that basically feeds the immune system with endogenous levels of antigens. And we hope, of course, to show that that also leads to, in the end, broader and better control, immune control over the disease.
Well, and speaking of your lead candidate, it's now in phase 2. You're starting to see some positive results. What's your strategy going forward in terms of optimizing the clinical development as well as the eventual commercialization of the candidate?
Well, I think the most important thing is that we first wanted to do a monotherapy study, right? Because that's the only way to show that what you are seeing in the clinic can be assigned to your product. The reason we could do it as a monotherapy study is that there was really nothing else out there for those patients. Now, of course, oral azacitidine is on the market, so that will create a new situation, and that is what we need to anticipate for. So we have done work with preclinical models, including animal models, to show that the drugs can be combined. So I think to focus on that setting first, being the current patients that we are focused on, current patient group, but then in combination with azacitidine, that will be a very logical next step. But also, of course, we are looking into alternative patient groups, for example, a bridge to transplant or post-transplant patients that also still have MRD And then there's also a new group of patients arising because there's a new drug on the market called venetoclax that is also resulting in more patients achieving complete remission. Because venetoclax is still quite toxic, that is also a patient group we believe that we can, in the end, treat with the vaccine. So, in short, there is a very broad AML maintenance field coming to the surface. We are currently positioned, we believe, in a very specific patient group, but also with a very clear way to move the drug forward. But longer term, we can look broader into how to position the drug in the AML maintenance field.
And finally, what are some of the milestones you're most looking forward to in for the rest of 2022?
Yeah, I think the most important one is, of course, the AML data. It's our most advanced program, but also we start to see now that the data start to look very promising. But to really deliver those data by year end, that give us the confidence level that we can be very competitive in this landscape. That is, I think, first and foremost what we need to achieve this year. And after that comes everything else.
Great. Well, thank you so much for your presentation today and answering the questions. And we look forward to continuing to follow Immunicum in the coming year.
Thank you, Michael.